Technique of raising tall seedlings. Importance. Production of tall seedlings has been an area which received considerable attention during recent years. Tall and sturdy plants, when planted, establish with early rains and withstand drought. At present, it has been thought mainly for aesthetic forestry, particularly avenue plantations, creation of parks, village wood lots, sacred groves in temple lands, in rural areas, and such other aspects. Tall seedlings are not prone for damage by animals because of the height of seedlings with highest percentage of survivals. In first year of planting itself, the greenery will spread in the area. Stages of raising tall seedlings. 1. Seed collection. 2. Preparation of primary bed. 3. Transfer to 4 by 7 inch bags. 4. Transfer into 14 by 20 bags. 5. Debudding. Seed collection. In order to produce of such tall seedlings of more than 2.5 meter, care has to be taken with regard to seed collection, container, soil mixture, primary nursery stock, etc. Generally, seed from trees of various species can be collected from February to June months. The important species that can be used to raise tall seedlings are Isidioracta indica, neem, Ficus religiosa, peepal, Ficus bengalensis, banyan, Ficus infectoria, Ficus mysorensis, Tamarindus indica, Anthocephalus cadamba, Terminalia arjuna, Eugenia jambulana, jamun, etc. The seed of the above species can be cleaned and kept ready for sowing. Raising of primary beds. Raised beds of size 40 by 4 by 1 are to be formed using soil mixture of red earth and farmyard manure in 3:1 proportion. These are called primary beds. The specified quantity of seed is to be soaked in fungicide solution prepared by mixing dithane M45 or Bavistin at 2 grams per liter of water for 1 hour. This eliminates inherent diseases in the seed. The dried seed can be sown in the beds. Transfer into 4 by 6 bags. The elite and vigorously growing primary seedlings of size 5 cm to 6 cm can be selected through culling out inferior seedlings and transplanted into 4 inches by 7 inches poly bags filled with 3:1 red soil and farmyard manure mixture. The beds arranged with 4 inches by 7 inches bags are called secondary beds. These seedlings are to be allowed to grow 8 inches to 9 inches in the secondary beds. After attaining height of 5 to 6 cm in primary bed, the tender plant are transferred into 4 inch by 7 inch bag plants carefully. At this stage, elite bag plants with vigorously growing terminal bud are to be selected and kept ready through culling. Transferred into 14 by 20 bags. 14 inches by 20 inches HDPE bags are to be filled with red soil and FYM mixture and arranged in one bag, rows of 50 or 100 bags in each row. The spacing between rows must be 50 cm to 75 cm to allow good aeration and light and also to facilitate watering. The selected bag plants should be transplanted into 14 inches by 20 inches bags so arranged. Debudding. After the seedlings attain a height of 1 meter, in big bags, auxiliary buds start emerging from axil between stem and leaves of the plants. Normally, the growth hormones or auxins flow from root system through stem to terminal bud of the plant. During the process, the auxiliary buds grab the flowing auxins and grow into side branches. In this process, the auxins reaching to terminal bud of plant gets reduced, thereby hindering the height of the plant. In order to prevent the auxiliary buds receiving the auxins, the auxiliary buds are to be nipped or removed when they grow to a length of 0.5 cm to 1 cm. Debudding process allows more auxins to reach the terminal bud to hasten the vertical height of the plant. Care is to be taken not to remove the leaf under the auxiliary bud while removing the bud. The presence of more leaves along the stem will increase the photosynthetic activity and produce more food material for the plant. The process of debudding may be stopped after the plants attain 3 meter and allow branching and crown. Debudding process should be continued periodically till the plant attains the required height of 2.5 to 3 meters. This takes 9 to 10 months' time depending of species. Each row of 14 inches by 20 inches bag plants has to be provided with support to plants by fixing 3 vertical casuarinous poles of 8 to 10 feet at both ends and middle and horizontal bamboo splits to provide support to plants as they grow. The bags are to be observed for roots striking the ground and shifted and placed on ground without cutting the roots. As an alternative, a poly sheet of 500 gauge of 50 centimeters width can be spread at the bottom of bags to prevent roots striking the ground. Conclusion. By planting such tall seedlings, good early results were obtained in drought-prone Anantapur district in avenue plantations, village wood lots, institutional planting, sacred groves, etc. An innovative technology of SWAR is piloted in 2017 under the MORD-GIZ Environmental Benefits Project in MGNREGS, AP. It demonstrated transformative water saving in irrigation with rich plant growth and yield to fruit farming in the drought-prone areas of AP to be successfully scaled in 2018 with government agencies, institutions, NGOs and farmers.